Welcome back friends. Good evening. In this video we are going to see about packaging of DNA helix. So about the DNA helix I have told you in the previous session. So the DNA helix consists of base pairs. So I have said about the distance between the base pairs and the distance between the width of the DNA helix, etc. Some things I have been told in the last session. So in this session we are going to see about the packaging of the DNA helix. So what will be packaging of the DNA helix? So the DNA will be having very much length here. Actually in humans as well as bacteria, the DNA length will be very high. So this much length will be compactly packed and they will be placed inside the nucleus in case of eukaryotes. In case of prokaryotes, it is called as nucleoid. So how they are packed? So before going into that, you should know about the length, how we can measure the total length of the DNA helix. For example, if you take human beings, we can have millions of cells. So millions of cells, when each cell consists of 46 chromosomes. So 46 chromosomes, where it came from? 23 from mother, 23 from father. So totally 46 chromosomes are there. So all the 46 chromosomes we have to measure actually in single cell. So in single cell consists of 46 chromosomes. So 46 chromosomes, what will be the length of the DNA helix in a single cell? First of all, you have to measure that. I have given here example. So total length of the double helical DNA. So total length will be measured with the formula. So what is the formula? You can see here, total number of base pairs into distance between two consecutive base pairs actually. So what do you mean by total number of base pairs? So total number of base pairs means in an individual cell, how many base pairs are there? For example, AT, GC, CG, like that. How many base pairs are there present in a single cell? So that is called as total number of base pairs. So in case of humans, we have, you can see here 6.6 into 10 to the power of 9 base pairs. So humans, as we are diploid organism, so diploid cell consists of 6.6 into 10 to the power of 9 base pairs will be there. So this is the total number of base pairs. So into distance between the two consecutive base pairs. What do you mean two consecutive base pairs? So in the last session I have said during the study about the structure of the DNA, between the first base pair and the next base pair, that is consecutive base pairs like this. Suppose A and T is Z, GC is Z. So between these two, so what is the distance I have told in the last session? So it is 0.34 nanometer. What do you mean by nano? 10 to the power of minus 9. So we can write it as 0.34 nanometer. Instead of nanometer, you can write it as 10 to the power of 9 meter. So this is about this total number of base pairs and distance between the two consecutive base pairs. So how we can measure the length of the DNA means with the help of the formula. So formula is total number of base pairs into distance between the two consecutive base pairs. Now we can easily measure the length of the DNA which is present in an individual cell. So for in case of humans, so we don't know what is the length of the DNA present in our human cell. So we can take it as X. So X means unknown, we don't know. So equal to total number of base pairs. So we have how many base pairs? In single cell we have 6.6 into 10 to the power of 9 base pairs. So into, so distance between two consecutive base pairs. Consecutive means between this and this we have what? 0.34 nanometer. So we can mention it as 0.34 into nanometer. Instead of nanometer you can write it as 10 to the power of minus 9 meter. So it is for per base pair. For single base pair we have this. So by divided by base pair you can write like this. So when we simplify this, you can cancel this base pair and this base pair we can cancel. So after cancelling, then you can see here plus 9 to the power of plus 9, here to the power of minus 9. So plus 9 minus 9 we can cancel this also. So what happens? So base pairs also we can cancel. This also will be cancelling. So what we will get? 0.34 into 6.6. When you multiply this, you will be getting the answer 2.244 here meter is there. So meters. So approximately we can tell in single cell of the human, that is mammalian cell, you have 2.2 meters of DNA. So the length of the DNA in a single cell will be 2.2 meters. Similarly, in exams also they will be asking, in NEET also they will be asking. In case of NEET, they will be giving some different values. So they will be asking to calculate your length, or they will be calculating this total number of base pairs, or maybe distance. They will be asking to find any other thing. So you can easily identify with the help of this formula. So again I will be one example. So in this case they have given the total length of the DNA in Ecollege. So Ecollege consists of 1.36 mm. So in this case m means what? 10 to the power of minus 3. So you can write it as 10 to the power of minus 3 meter. So here what you have to find? Total number of base pairs we don't know actually. So we have to find that. So total number of base pairs we can take it as unknown, that is x. So into, so for all organisms I said, from bacteria to higher animal like human beings, the length between two base pairs will be same. It will not change at all. So you can check it as 0.34 into 10 to the power of minus 9 base pairs. So this value will not change. So this will be the constant. So you can take it like this. So keep x one side and bring all the things to one side. So you can get here 1.36 into 10 to the power of minus 3 meter. So divided by, so the 0.34 will go here. It will be divisible here. 0.34. This minus 9, when it comes to this side, it will become plus 9. So plus 9 and minus 3 will be what? So 9 minus 3 will be 6. So it will be 10 to the power of 6. So x equal to 4 into 10 to the power of 6 base pairs. When you divide 1.36 by 0.34 gives you the value 4. So into this plus 9 minus 3 gives you 6. So you will be getting 4.4 into 10 to the power of 6 base pairs. So this is the number of base pairs present in Ecollege cell. So this way only you have to identify the length as well as total number of base pairs. So this will not change. This will be a constant. So keeping this as constant, you can substitute and you can easily identify the values. In case of NCERT as well as in the case of NEET also, you can easily identify the length of the DNA. Now coming into the concept here, you can see here 2 into 244 meters. You can imagine the length. 2 meters will be how much you can imagine. So when you take the cell, inside the cell, nucleus will be a smaller one. So inside nucleus, if you take the nucleus, what is the dimension of the nucleus? So dimension of the nucleus is 10 to the power of minus 6 meters. You can imagine. So within this small nucleus, 2 meters length, the DNA has to be compactly packed inside this nucleus. How it will be packed? So that is one mechanism how they will be packed. So I will be discussing about that in the upcoming session. Thank you friends. Thank you for watching this video.